Thank you, Belal. So I also start with just giving a couple of slides on the plant side of things. I know many of you come from a medical background or pharmacy. I've been talking to a lot of students that are studying pharmacy, studying medicine. There is a plant component research in ICGEB, and we know that on many ICGEB member countries, agriculture plays a fundamental role in their economy and in providing food also for its people. So we do have several plant research groups, and we intend to increase them also in the future. You see here we have the plant biology group here. Plant biology groups we are only in Trieste and in New Delhi. New Delhi is a lot stronger in plant research than Trieste, and also we have recently, there's also somebody in Cape Town that's starting to look at biopesticides as well. And there's also a group doing industrial biotechnology, a major group in India. I'll be talking about that this afternoon when we discuss technology transfer, because they're mainly involved in biofuels. So, and that is very much linked to agriculture because they use agricultural waste. Try to optimize the use of waste and turn them into biofuel. Looking at plant biology in general, I just have one slide. We have several fields of research. One is plant transformation technology, and we have a group that is doing leading work on transforming plants and inserting genes in chloroplasts. So having high level expression of genes using this technology. There's also other groups that are looking at using CRISPR-Cas to provide herbicide tolerance, and that is very interesting, because using CRISPR-Cas then you can also avoid the GMO strain gene regulatory network just by changing the endogenous genes of plants and making the encoding products, the enzymes, resistant to certain herbicides. There are groups looking at improving the ability of micronutrients. There are plants do have certain activities and certain proteins which make certain nutrients insoluble. So by engineering those pathways, we can then make the nutrients more available to humans when they eat them. Okay. And then also there's very interesting plant pathology studies. So, the compatibility and incompatibility of pathogens, it's a very important questions that are studied in many diseases. And scientists are studying using an insect. And what they're finding is very interesting, that actually through salivary glands, the insect injects some proteins that attenuate the immune response. So scientists are looking at those, trying to understand how that takes place in order to make the plant more resistant to the insect. And then also looking at microRNAs in biotic and abiotic stresses, and looking also at salinity and drought stress by looking at certain pathways that are important in providing more stress to these conditions, which are very, very important. Salinity stress is a major concern in the future, because we are turning our soils more and more saline. So we need to grow plants in much more saline environments in the future. So there is a program in plant biotechnology, and also plant basic research in ICGEB. So for any of you who are interested to pursue, interested in these topics, I can provide you the contacts, and you can contact the PIs to initiate collaborations or applications that we discussed earlier. Coming to my work, I also have the privilege to an opportunity to talk to you about my work. I have a bacteriology lab in Trieste, and my interest for many years has been cell-to-cell signaling in bacteria. And in the early 90s, scientists were beginning to discover that bacteria, even though they are unicellular organisms, they produce small signals. So when I say signals, it's more chemicals, or sometimes even very small peptides, that they use as their language. So these signals are secreted into the outside, and the microbial community can detect these signals not only which signal is there, basically which molecule is there, and how much of it is there. And those are two very important pieces of information for a bacteria, because the type of signal will tell the bacteria what kind of neighbors are around, and how much of it there is will also tell them how many of those bacteria are around. So that created, really opened a new field of research, which a lot of people, including myself, joined and started looking at which signals are being made and what is the function. Because what these signals do is they allow a community to synchronize their behavior. So when there's a lot of that signal and that specific signal, that means I have a lot of next of kins. So I have a lot of very close relatives. So, and there's a lot of us, we start doing things that we would not do when there's a few of us, like attacking a host, like monopolizing a nutrient, like making some beneficial interaction with plants. So a lot of these community behaviors have been studied in so many different bacteria, living in so many different niches, beneficial bacteria, pathogens. So basically what I'm saying is that bacteria do not want to live alone. They want to live as part of communities. And since they want to live as part of communities, they need to understand, they need to communicate with their neighbors to know what they have to do and to behave as a community. So I've studied many years that, and I've studied them in a really simple setup. So I use plant-associated bacteria as my models. And I grow my bacteria in sterile conditions in my conical flasks, and I grow them, and I do some chemistry to see what signals they're making, and then I do some genetics to see which genes are involved in making the signals and responding to the signals, and then I understand the system and what the system regulates. But I now stop that because I realize how far away it is from the wild. Because if we go to the wild, especially to plants, and we look at microbes in plants, the communities are very complex. We don't find, there are with very few exceptions, we don't find that a niche near plant is colonized by a pure species, if not a pure strain. There are very complex mixed communities. So my challenge, myself and a lot of other colleagues of mine, we need to study signaling in multi-species communities. What happens? Because all these different species maybe make different signals. They need to understand, they need to understand who are the cooperators, who are the competitors. So there's a lot going on in these communities in order to establish a stable and successful community. There's a lot of communication going on. So that's what I want to study. And this is what I've just told you. I've been studying signaling. So bacteria produce signals, and these signals. A community responds to the type of signal and the concentration of signal, and then they will respond by expressing genes in response to these signals, which are beneficial to the community. Bacteria are social organisms. 4 or 5 years, a new term was coined, which is called social microbiology. So there are scientists out there that study social microbiology in order to understand also social behavior. And also transfer this kind of studies to also humans and other animals. Because bacteria produce a lot of things that they throw out to the environment. So as soon as you produce something and use your energy to then produce something that you throw out to the environment and make it public, make it available to everybody, automatically you're a social organism. So when you do that, you want to make sure that your neighbors are friends. Because if you work hard and use your hard-earned energy, your metabolic energy, to throw out something that your competitors will use and then kill you or remove you, then you're doing a bad job. So this social microbiology is a very new field which is creating a lot of interest and theories around it. And people are using bacteria to develop social theories, even used in economics, for example. So it's a new field which is because studying bacteria is rather simple, so you can use these social models to formulate ideas. But what I want to do now is looking at these interspecies scenario. What happens in these complex species? And that's going to be the challenge of the future. And we're finding quite a lot of difficulty, myself and colleagues, to study this and to use model systems to study this. And so I look at interspecies communities of bacteria which are associated with plants, both pathogenic and beneficial for different reasons. And, believe it or not, pathogenic and beneficial communities use very similar systems of communication, and establishment of communities, which also will have serious implications, because if we want to control a pathogen, we might, the strategies that we use to control a pathogen might also affect a beneficial community. Okay, so I'm particularly interested in novel signals, novel regulators, so the signal also has to be sensed, and also I'm using synthetic communities. I'm using different strains and making synthetic communities in the lab to try to understand this. Okay, this is just a flash of some of the languages of bacteria, some of the signals. And what I just want to show you here is that they're rather simple. I mean, I'm not a chemist, but chemists can very quickly synthesize a lot of these molecules. They're very simple. A lot of them are diffusible, so they move in and out of cells without any transport. Okay. Most of these are produced by gram-negative bacteria. You can see the names of the bacteria underneath. I mean, these guys are very serious plant pathogens. These guys are produced by beneficial and pathogenic protobacteria. You know, this guy here is produced by, actually produced by both gram-positive and gram-negative. In fact, is believed to be produced by every bacteria, which makes it interesting, because what scientists think is that since it's produced by every bacteria, is a kind of cue for the bacteria to just understand what is the bacterial load around it, just the general load around the bacteria. So, and what I also want to tell you, that many bacteria produce more than one single signal. Just like humans, some humans can talk more than one language. A lot of bacteria can also produce more signals, so can speak more languages and communicate in a much more intricate way. So I've been focusing a lot on this language, because it's actually, myself and many colleagues use this language because it's very easy to study. So at the moment it's the most common language, but I think it's the most common because it's the easiest to study, easiest to detect a signal. There's a lot more signals out there that we haven't even found. We are really the tip of the iceberg in terms of understanding bacterial communication. And this is a very simple molecule. It was first discovered, it's the first signal discovered in bacteria. It's called the Homoserine lactone, and this is the first one actually discovered from a marine bacterium, beneficial marine bacterium. And this is a 3-oxo, it's called a ketone, carbon-6 Homoserine lactone. And what you have so many different dialects to this language. You can have C8, C10, C up to C carbon 20, and at position 3 you can have different substitutions. So basically you can have so many variations but of this type of molecule, so many different dialects, which gives it specificity to certain bacterial language, you know. So if I speak that, if I produce this particular molecule, I will not be able to, you know, my neighbor bacteria maybe produces a 3-oxo-C8 and is not able to understand my language. So it does give some specificity to this family of to this language. So there's a lot of dialects which are very specific. And the system is so simple, you know, so powerful. So you have basically this signal here, this particular signal, but this can also be applicable to other signals. So you have basically inside the bacterial cell a protein, which is usually a homodimer. A for activator, R for repressor, is a very related protein, but this acts as an activator and this acts as a repressor. So basically when you have a low amount of signal, there's no binding. When you have a high amount of signal in the activator, the signal binds one-to-one, one polypeptide to one signal. And this binding is very strong. You create a homodimer, it will bind to a promoter and activate and start the RNA polymerase and transcribe the target gene. So that gene or genes which are regulated by this system, community genes, you know, genes which products are necessary for the community, you know, proteins which are secreted, molecules, antimicrobial compounds, virulence factors, so on and so forth. In the repressive scenario, you have a repressor, very related protein that will bind to the target gene and repress its expression. And then you have the opposite scenario, that high cell density, high concentration of the signal. So when there's a lot of signal around, a lot of your friends, then that will really, the repressor, homodimer will bind again. The homodimer will bind to each polypeptide, will bind to one signal, and that will relieve repression. And in this case, RNA polymerase can bind and transcribe that gene. This is much more common than this, but both systems are there. Let's skip this. This system, this kind of language that I'm studying, these Homoserine lactones have been discovered in so many different bacteria. And actually, what makes me happy about this slide is that the fundamental studies, the textbook studies of bacterial communication have been done using plant-associated bacteria, not the human pathogens. Then they came after. Probably, as you can see here, these are the underlying, the fundamental studies, the Cell and the Nature papers, have all come from these of bacterial communication have come from plant pathogens. These are very important plant pathogens. And then the Pseudomonas aeruginosa is probably the most studied bacteria right now for bacterial signaling because it's an opportunistic human pathogen, especially in immunocompromised patients suffering from cystic fibrosis. They cause chronic infections. This bad guy is 4 or 5 different signals that are produced, 2 of which belong to the Homoserine lactones. So, and a lot of the beneficial guys as well produce these kind of signals. So as I was saying before, here we have the plant pathogens and the plant beneficial using a similar language, different dialects. So this, unfortunately, I have no time to talk today, but fantastic scientists have found ways to block the signaling. Unfortunately, the blocking is not dialect specific. So by using these, it's a bit like taking an antibiotic. When we have an infection, we take an antibiotic and we affect our commensal microbiota, right? Which is, we cannot take an antibiotic forever, can we? Because we're sick. Same with this system. There are beautiful ways the scientists have found to block this communication, so affect bacterial pathogenicity, because if the pathogen cannot communicate, it's much less pathogenic because it does not attack the host as well. But unfortunately, that blocking system will also affect beneficial communities that live with plants, which are many, as I'll come in a section. So one thing that we discovered that we were lucky, because, you know, in research, you have to be lucky, you know. It is true, you have to be good, but if you're good and not lucky, you know, it's difficult. So we were lucky that we discovered something, you know, approximately 19 years ago. We were looking at rice pathogens and rice beneficial bacteria. So because I use rice as my model plant system for obvious reasons, it's the most important crop in the world, by far. So we were looking at beneficial bacteria and pathogenic bacteria, and we were looking at this language. And we were asking ourselves many years ago, I don't know, 9, 10 years ago, do they make this? Do they talk this language? And if so, if they speak this language, what do they use it for? Very simple questions, actually. And, you know, in bacteriology, it's very simple research. You can make knockouts, you can purify signal. You know, the genome editing in bacteria, we've been doing it for 20 years, you know, even more. So we can move quickly if we find interesting genes. But what we found, which was new, was that we found bacteria that could not speak the language, so they did not produce a signal, but they had the protein to respond to the signal. So it's like listeners, but not speakers. So we thought, that is very interesting. How can we why do we have bacteria, they don't speak, they don't talk to their neighbors, but they can listen to them? Right? So we found proteins in the genomes. This was started before the genomics era. We cloned them first, and we found proteins that had, sorry, this is actually missing some stuff. This is the N-terminal, this is C-terminal, it's about a 250 amino acid protein, a modular protein with 2 domains. We found this protein having a domain that binds the signal, this kind of signal, these Homoserine lactones, and a DNA binding domain that binds the promoter. This then makes a homodimer, as I've shown you before. So we found bacteria that have this protein. So we had models. Wow, we have bacteria that can listen to neighbors. Right? And then the scientists in the US were reporting also that plants were making signals that are very similar to these Homoserine lactones. So it could be that the plant was also producing signals that the plant could detect. So we said, oh, maybe the plant, maybe it's not responding to bacterial neighbors, but to responding to signals from the plant. So I don't want to get into too much detail, but we studied this and we made some nice discoveries, I think, anyway. And we've shown that these, for example, I work with Pseudomonas genera, which is very abundant in the rhizosphere, so in the soil closest to plant root, which is actually the gut of the plant. If we think about the human gut, which is so important for our health, the equivalent in plant is the rhizosphere, which is the soil closest to the plant root. the plant exudes 15% of the carbon and nitrogen that it produces to recruit a microbiome, a microbial community next to the root for its health, which is very important for the health. I'll show you a slide in a second. And what we found in these Pseudomonas, that we sequenced about 50 genomes of these Pseudomonas, and then we looked in the data banks, because now the microbial genomes is going crazy. I mean, there are hundreds of sequenced every month. We looked at Pseudomonas that live in the rhizosphere of plants, which are, Pseudomonas are a very good colonizer of rhizosphere of plants. We found that 82% have a gene protein for listening to neighbors, and they don't produce signals. So we're studying these proteins, and we classify these proteins into nine subfamilies, and we believe that these proteins, I cannot give you, we're looking at I have a PhD student, very good PhD student, looking at this right now, making knockouts, looking for supernatants for signal, understanding to which molecular response. But what we think is that these proteins, gene proteins, are very important for interspecies signaling. So our hypothesis is that bacteria, apart from speaking their own language, they have the ability to listen to languages from other guys, and this is a very important family of proteins that listen to neighboring languages, and very important for living in an interspecies bacterial community. Okay? So I'm talking today mostly of things I'm actually doing right now, rather than give you an historical account of what I've done in the last 15, 20 years. So I'm talking about the research that I'm actually being involved right now, because maybe I can find some collaboration. Some of you are interested in what I'm actually involved right now, rather than give you an account of what I've been doing, giving you stories of what I've been doing in the last years. Okay, so this is one thing that we're doing, and the last 10, 15 minutes that I want to talk about is, NGS has really created a revolution. I mean, now we can sequence. When I did my PhD, I sequenced 6 genes without oligonucleotides, just by cutting fragments, making small libraries of 1 kb fragments, cutting them with 3 different enzymes that recognize 4 base pairs. It was an incredible amount of work, all by hand. The first genome project funded by the European Community sequenced Bacillus subtilis. It involved 50 labs in Europe, and it costed the equivalent of €1 million today, but back in 1995, so who knows how much money that is now, all done by hand. So if somebody came to me when I started my PhD and says, "Oh, don't worry, Vittorio, 20 years time you can sequence a bacterial genome with €100, I thought he was going to be crazy. Now it's even less. So now what we are able to do through NGS, we can photograph complete microbial communities through microbiome studies, by doing amplicon 16S sequencing, I'll show you in a second, or by doing metagenomics, by sequencing everything. So that now we can understand the complete microbial community. Beforehand, we had to take the rhizosphere or the roots and grow every single bacteria that we could grow, and then see what it is, and then try to understand the community. And keep in mind that we can only grow 30–40% of the bacteria that live with plants. The others, we just can't grow them because we don't know how to. So that will give you a really bland picture as well, and it will not give you the abundance either. So NGS really created a big possibility for us to try to understand microbial communities, right? And to try to understand who's there and how many are there. So the plant microbiome is I don't have to tell you, but a lot of studies have been done on the human microbiome. The human microbiome research is so far ahead the plant microbiome, for obvious reasons. In North America and in Europe, funding for medical research, it far outweighs the funding for agricultural research. In Europe and North America, we don't give a lot of value to, in my view, not as much value to agricultural research. We have supermarkets everywhere. Most people have problem with cholesterol and so on and so forth. So why fund research in agriculture? Whereas, in our ICGEB member countries, there's a lot more need for agricultural research. So the plant microbiome research is now catching up. So a microbiome is the complete microbial community that lives together with a specific niche. So the plant microbiome is the complete microbiology of a plant. And just like in humans, we have 10 times more microbial cells living with us than human cells. Same with plant. And there's a lot of bacteria that live together with plants, and these are determined, very important drivers of the microbiome are the soil type, the host genotype, and the vertically transferred. So the seeds that we plant, the farmers, the crops, the seed of their crops, already the seed inside as a microbiome. That will be very important for its health. So these are the drivers, you know, the soil, the host, and the host genotype will recruit most of its microbiome from the soil. So the soil is by far the most important determinant for the microbiome of a plant, of a crop, of a tree, of any, okay? So, and again here, we have to be careful. Just like humans, we have the oral microbiome, we have the intestinal microbiome, we have the skin microbiome. In a plant it's the same. We have the rhizosphere, which is like the gut. That's the closest to the plant root microbiome, which is really determined by root exudates. You know, and this is a research which is so behind, but we have to understand how the plant signals to recruit a healthy microbiome. The phyllosphere is anything, all the surface structures, aerial surface structures. Endosphere is what I'm very interested in. The inside, so bacteria which mainly from the root get inside the plant and then even move upwards, so they live in intercellular spaces inside the plants. That is very interesting because that's where the pathogens, a lot of pathogens are. And here we have good beneficial bacteria that live in the same places as the pathogens. So it's an excellent model to keep the pathogens away. And then, as I said, the seed microbiome. So these are the four. If you look at plant microbiome papers, usually they will focus on one of these four. Some really rich labs will determine all four. The reason I say that, because when you do microbiome research, you need a lot of samples to do good statistics. So you usually, in papers, you will see that scientists are focusing on one of these areas, just like human microbiome scientists focus on lungs or oral or respiratory tract or gut, skin, or so on and so forth. There's a lot of different areas. And the reason the microbiome functions for a plant are so important, and there's a lot of good evidence for this. You know, efficient nutrient uptake, just like in humans. Abiotic stress, surviving a stress, the microbiology plays a fundamental role. Plant growth promotion, there are bacteria out there, and mainly bacteria, but also some fungi that produce plant growth hormones, cytokinin, auxins, which determine root growth. They're not useful for the bacteria. They're useful for the plant. Of course, the plant gives something back to the beneficial community of the roots. Inducing plant immunity. So plant health in general and disease resistance. This can take place in two ways. Just by niche exclusion. If I can colonize very well a niche and there's a good networking and a good microbiome is very strong, the pathogen cannot establish. They keep it away. Alternatively, there's a lot of microbiome members that produce antimicrobial compounds, antifungal compounds, antimicrobial compounds, so that when the pathogen arrives, the community survives because it is resistant to the compound, and they kill the pathogen. Okay? So there's a lot, you can immediately see a lot of applications. And what we're doing using rice, I'm putting my money on this and I hope it's good. As I say, I'm telling you the work I'm doing right now, is that we're looking at certain rice diseases, and we're looking at disease plants, and in the same field we're looking at healthy plants. So for the disease plants, we determine the pathobiome, because again, this is also a new term which has been coined maybe 4 years ago, the pathobiome. When we look at disease of plant and we go to the infected site, and we look at the microbial community, you think, oh, the pathogen is there. Yes, the pathogen is there, but it lives together with a lot of other microbial members. So what happens there? And I've published several papers to show that there are cooperators. So the pathogen will cooperate with some members of the microbiome. And they will both benefit from this cooperation. So it opens your eyes a bit to the pathobiology of the disease. So we have to think also that the microbiome will play a role in the disease. And then we're looking at the microbiome of healthy plants, and then we can compare the two and say, what's the difference? And maybe get information about cooperators of the disease or antagonists of the disease. And then think about signaling, think about also control, bacterial biocontrol of the disease. So both fundamental science and application. And don't worry, I'll 10 minutes and I finish a few slides. And here, just to show you where ICGEB comes in. You know, the privilege I have of working for ICGEB, because, for example, I'm looking at 3 rice diseases: sheath rot, foot rot, and leaf blight. And these are, for example, this is very serious in Italy. Italy is the first rice producer in Europe, which is, you know, Europe is small, 250,000 hectares, which is really not significant if you think that in India or Thailand or Vietnam, the acreage, hectares in Vietnam are much higher. But nevertheless, this is a very serious disease. Foot rot is, at the interface between water and air, this air rots due to a bacterial decay there. The bacterial pathogen will infect and will colonize and will rot. Rot, why? Because it gets food, right? Rotting. This is another rot, but it rots the sheath covering the panicle, and this is very serious in Africa, especially when you grow rice at high altitude. And the rice, well, this is the second most important pathogen of rice. It's devastating in Asia. It's classified also as a bioterrorist agent. This is a very serious disease, Xanthomonas oryzae, and this is a vascular disease. So this guy from the leaves gets in through the hydatodes where the water gets in and colonizes the vascular system and blocks it. So you can see chlorosis, the leaves turning yellow, and slowly they can kill the plant. Very serious. 10% of rice worldwide is lost due to this pathogen. So you can imagine the research going on in this pathogen, especially in Asia. So what we did, we went out and collected samples of sick plants, as I showed you on this slide, sick plant, healthy plants, and compared the pathobiome and microbiome. But appreciate the work. I had to collaborate with the International Rice Research Institute to get the samples, with the National University of Vietnam, with the Italian Rice Association, and all of this has been done properly with material transfer agreements, following the Nagoya Protocol. We had to sign all the paperwork to get the samples, and going out there, getting the samples, getting the DNA to Italy. It took about 8 months. But thanks to ICGEB, I could get this, and I'm collaborating with Outstation of Italy in Africa, and with collaborators in Vietnam. And also, I'll show you next slide, I'm also collaborating with other countries as well. And we did all this work, and we did all this pathobiome, microbiome. And just to show you a few slides, for example, this is all unpublished data with the pathobiome of the Italian disease in the sheath rot. And when we look at the pathobiome of the sick plants, we see the pathogen. This is. So what we do here, we take the infected samples, and at the same field, the healthy samples of the same area of the plant, we take that plant area, we purify total DNA, we amplify the 16S, which is a taxonomically important gene, the 16S, ribosomal DNA gene. We amplify it, we get hundreds of thousands of amplicon, we make an amplicon library, and then we send it for NGS, and we get millions of reads, and we get the reads back, and we analyze the reads. And then we make a picture of the community, who's there and how many, based on the sequence of the reads and how many reads we get information on who's there, who's in that sample, which bacteria in that sample. And these are the results. So these are the number of reads. And this is the number of reads of decay of the pathogen. And you can see when you look at the infected samples, wow, it's crazy. There's a lot of pathogen there, of course. We infected samples. But if you look at the same plant, healthy plants on the same field, the pathogen is not there. So wow, that's the first thing. Okay, not a big surprise. But then we compared the total bacterial population of the healthy and the disease, and we start comparing them, and what we see that there are clear enrichment of general bacteria in the sick samples. And we analyzed over 150 samples, so statistically very strong, that these guys here are enriched, and in my view, these guys cooperate with the pathogen in the disease process. And in the same time, we see a big enrichment of bacterial general in the healthy samples, and these guys could be involved in keeping the pathogen away because these guys are healthy. And maybe the microbiome of the healthy plants is much stronger, and decay comes and cannot infect. And here you have this, this is a very nice, differential analysis. And you can see here, these are the statistically, this is decay, and these are the statistically significant genera which are enriched in the sick sample, and these are the ones that are enriched in the healthy samples. And then I don't want to spend too much time, but then we go back to the samples. We go back to the samples. We isolate these guys because we kept a sample at -80 of all the samples we analyzed by microbiome, 16S amplicon. We keep a sample in the -80. We can go back to it. We keep a part of it in -80, And then we're isolating these guys, and what we're showing is that these guys are actually, we isolate, so it's a lot of work isolating. Then you have a lot of bacteria, you have to find the ones that you want, and then you do co-infections, and we see that in the co-infections, together with the pathogen, when you infect with both of them, the disease is stronger, and the putative cooperator actually grows much better. So there is, in my view, and now we're looking, there is communication and interaction between the pathogen and these possible cooperators, and that's what we're studying. Okay. And again, I just don't want to, this is just repeating, this is the other disease in Africa. We went to Burundi to collect samples of the sheath rot, and again, disease, we see a lot of this is called by the pathogen Pseudomonas vaginae. We see a lot of the pathogen, we don't see in the healthy samples. And in this case, the collaboration with the International Rice Research Institute, they want us to focus on controlling the disease using biocontrol agent, biopesticides. And we're now actually, the PhD student in my lab from Burundi is now actually in Burundi, and he's isolating all this analysis. Showed us that these guys, Sphingomonas genera, so bacteria belonging to Sphingomonas, Sphingobacterium, Pantoea, Hymenobacter, are very abundant in healthy plants. So he's isolating members of these guys, and then we're going to test them whether if we can use these wild types, of course, and spray them on rice plants, is it more resistant to the disease? And if it is, then we might produce a product that we can give to the farmer and say, well, you know, at that certain age of the rice, when the infection is likely to initiate, to establish itself, spray these bacteria, this mixture, and your plant might actually keep the pathogen away. So it's like controlling the disease using microbes, wild-type microbes, so we don't have to use chemicals. Right? Okay, so to finish up, I just have 2 slides, so 2 or 3 slides. To finish up, I also want to show you, because I'm proud of this, because of ICGEB, I'm also looking at the rhizosphere microbiome of coffee plants, because in Italy, in Trieste, we have a very important coffee company called Illy, and they contacted me and says, why don't we look at the gut, the plant gut, so the rhizosphere, and look at the microbiome of the rhizosphere of Coffea arabica, the high quality cultivar of coffee in different parts of the world. Again, look at this. I had to collaborate with, we did it in Burundi, we did it in Ethiopia, we did it in Colombia, Ethiopian Biotech Institute, Senicafe in Colombia, in university. This time we did it with the University of Bujumbura in Burundi, and this small project, pilot project, was funded by Illy. But again, can you imagine all the paperwork, talking to the people, signing the MTAs, getting the, you know, and we have people in these countries. People in my lab went to Ethiopia, to Colombia, you know, purifying, getting the samples, purifying the DNA, and then the DNA was sent to Trieste, and we did the Amplicon library 16S and determined the complete rhizosphere microbiome. And we're now analyzing it. And the objective here is trying to look for keystone genera, because in microbiomes there are some bacterial genera which are called keystones. So they are in the center. If you think of a network, there are central nodes in the network. If you remove some nodes, peripheral nodes, the stability will not change very much. But if you take away a central node, you can cause a collapse of the network. So we're looking for these central keystone genera that will keep the network together, because then, by comparing in the same coffee cultivar in 4 different areas of the world, then we can maybe estimate which are the keystone genera, and maybe we can make a fertilizer, because we can say to the farmer, here, use this product with these 4 genera of bacteria, because those are so important for your rhizosphere microbiome. And if you give a lot of them, maybe the microbiome can form quicker and stronger. So that's the aim of this work, and we have all the data now, and we're analyzing it right now together with a bioinformatician which is hired in ICGEB, and we will have, as Lauren said, we will have now a group of bioinformatics in the future, which will be very helpful in, because you know it's a lot of computer work, and because there's so many samples, and then you have to do the differential analysis. But we're confident, because we're already seeing that we think we can pinpoint the keystone genera. And to finish up, I have just to finish. I'll talk about it a little bit this afternoon with technology transfer. We have initiated a big project 4 or 5 years ago on endophytes of rice. So these are bacteria which start from the soil, go to the rhizosphere, and they get inside. And because we think, well, as I said earlier, these guys go inside, they colonize the plant well, they're beneficial. We're looking at the beneficial ones, so we can then give them as products. So then we can enrich the endophytes and maybe make the plant healthier. And a lot of these endophytes also help nutrients, help the acquisition of nutrients. So we have, I mean, this is just appreciate. I mean, you can imagine isolating 1,300 bacterial pure cultures, sequencing them, doing the 16S. So we had a very big project funded by a foundation actually, and we got down to a bunch of endophytes which we think are good endophytes. So when I say good, I mean we purify them, we give them back to the plant, we see that they colonize the plant well, and they are plant growth, and they promote the growth of plants. So the plants are healthier when we inoculate them with these endophytes. We sequenced all the 48 genomes with a grant from the Joint Genome Institute in California, and now all these endophytes have been licensed to a big company in the US. So we have a research agreement, and now I cannot go into too much detail, but we're looking at these 48 endophytes to make a product for rice, endophyte product, so a product where bacteria will go through rice, get inside, and the rice will be healthier in getting nutrients and growing faster. Okay. So that's what so this is you know, and this is the last project is very applied. We have good contacts with the company. I'm collaborating with a company in the US. It's called Indigo, and also with a company in Germany called Matex, which I didn't put here. So we're also looking at applications, making, you know, developing biofertilizer for a more sustainable agriculture, because, you know, there is a world consensus that we have to reduce by 2022 the use of fertilizers by 10%, and these microbial kind of products are the most promising alternative. And this is currently my group, which is actually not updated now. I have a new PhD student from China, just joined my lab. But as you can see here, again, just like Lawrence, I have people from Peru, from Burundi, now I put Ikshu from China, and also and I have 2 research assistants which have been with me 20 years. So, and I have, you know, we are privileged to work for ICGEB, but if we don't find external funds, we're not able to work well. So you know, we spend a lot of time looking for funding, and these are the people that have helped funded my lab. Okay, thank you very much. Thank you. Please.